the kind introduction. And before I begin my overview of our services and products, I'd like to introduce our company quickly. So we've been operating out of a Malvern office for nearly 15 years, and originally we were founded as a company based on miniaturized high-throughput screening platform, but ever since we've evolved into this early-stage drug discovery CRO with emphasis on kinases and epigenetic targets. And we also offer cell-based assays, custom assay development, as well as protein production. We provide services to customers worldwide, and that include big pharmaceutical companies and smaller biotech companies, as well as academia. And then this year, we have been selected as a specialized center for the Chemical Biology Consortium. I'm very proud to be chosen alongside of 14 other great institutions and academic labs. Being a CRO, our main focus is obviously services that include mostly in vitro-based assays that cover different enzyme classes listed on the left. And then we have a very large kinase panel consisting of about 600 proteins, as well as many different epigenetic enzyme targets, as well as many others. Recently, we've been expanding our product line that consists of recombinant proteins, protein substrates, epigenetic inhibitors, and some kits. And then we also spend a small portion of our time working on internal research projects that are funded by SBI grants. And they mostly aim at discovery of probe molecules for bromodomains, HMTs, and demethylases. And that involves producing proteins, making assays, as well as screening compounds. So the field of epigenetics has advanced greatly over the past decades, and it comes from our understanding of epigenetic regulations of gene transcriptions at the level of chromatin organization. And then as we learn more, identify a lot of proteins that are involved in this regulation, specifically writers, readers, erasers of either histone, tail marks, or modifications of nucleic acids. And there's a clear association in disease, whether it's cancer, inflammation, or metabolic disorders. And then it's also evident from talks that we hear at these meetings that there's great potential of drugability and therapeutics in these targets. And then this research depends on availability of good epigenetic tool compounds and then assays and reagents. And that's one of the main goals of reaction biology, is to produce these quality epigenetic reagents, as well as develop biochemical assays for these targets and make them available for screening and profiling of drug candidates, and then in later stages make available cell-based assays for lead validation. So I'll just briefly mention what kind of protein products we have, and obviously I cannot go in detail about them. So main two classes are epigenetic enzymes and then reader domains. So as you can see from the top graph, we have a large collection of methyltransferases, histone methyltransferases, as well as DNA and other methyltransferases. And these include wild-type proteins, as well as mutations that are relevant for disease. And then, as you can see on the top right corner, we produce high-quality proteins either in insect cells or E. coli, and then purify them to at least 80% purity, and show an example for NSD2 on top. And then, if necessary for activity, we provide them as complexes. So, for example, MLLs and then PRMT5, MAP50 complex. And then also each batch is tested for activity, and then we compare it to internal standards to ensure that products that we sell conform to certain standards, and then they'll be working in your hands. And then for reader domains, our biggest product line is bromodomains. As well, we have a large number of chromodomains. And then we're actually expanding it right now to include different domains from demethylases as part of our SBR grant. And I'd like to take a minute to cover our bromodomain product line. So we were able to achieve over 85% coverage of bromodomain family. And then most of the proteins come as single bromodomains, and in some instances we have constructs that cover 2 bromodomains. And then we also have several full-length BRD proteins, including BRD4 full-length. And we also were able to make them with 2 different tags, his tag and GST tag, and that allows you to select what kind of assay you want to perform downstream. And then a lot of them are available as products, and then we also use them for internal research. And then having this great collection of proteins, we were able to put together a great selectivity profiling tool that we called BromoMelt, because it's based on thermal stability based assay. And it has a great coverage of bromodomain family, as you can see on the left by the tree, and then the circles indicate which proteins are included in the set. And then the table just lists the names as well. We have 77 proteins in the set, and it represents 56 members of the family. And this BromoMelt is available as profiling service at Reaction Biology, or you can do it in your own lab. All you need is a qPCR machine, and then every other component is included. When you get the plate, you can test each bromodomain at 4 different conditions. So you need a DMSO reference, then there's a control mix that contains 5 different molecules, the known compounds that bind bromodomains, and then your compound of interest can be tested at either 2 different concentrations or a single concentration in duplicates. So as the protein is heated, it denatures slowly, and then if compound binds, it will stabilize the protein and then change the melting temperature. So in a matter of about 2 hours, you can get individual melting curves for all proteins in the set, and then as illustrated on top by BRD2 family. And then also you can represent all the TM data as a compound profile, and it will clearly indicate the substrate selectivity for the compound that you tested. Last product line that I would like to mention is substrates for histone methyltransferases. So in the cells, these enzymes work in the context of nucleosome, but when you do these assays in vitro, they can accept anything from histone peptides to either native or recombinant nucleosomes. And some enzymes are more finicky than others, so it's important when you set up the assay to try different things and then select a substrate that will be most cost-effective for your application, as well as produce highest activity for the enzyme of interest. So at Reaction Biology, we produce everything but the peptides, so then it's available for purchase. Finally, I'd like to go over our assay services, where we can help you out at any stage from hit identification to lead profiling, and we put strong emphasis on data quality and on-time data delivery, as well as customer service. Any assay that we offer is compatible with screening, and then currently our maximum capacity is about 250,000 compounds per target, with a turnaround of about 2 to 3 weeks. And then we have 4 echo instruments in the lab, and it's a great tool to enable miniaturization because it delivers the compound directly into the well. And then you don't have to have a step of compound dilution and buffer, and you control DMSO concentration, since a lot of enzymes are sensitive to DMSO in assay. And then we did a couple of screening projects for our customers, and I cannot share that data, obviously. But recently, as a part of our bromodomain chemical probe discovery project, we conducted a screen for BRD4. So this came from the approach to find probes for different bromodomains to be able to set up FP assays. And if fluorescent polarization assay is quite very sensitive and easy to do and very cost effective. So we have currently a couple of probe molecules, and then here I show specificity for probe number 1 on the left, and you can see that it's mostly still selective for BET family, with few exceptions of binding to other subfamilies. And then we're currently screening for other probe candidates that can be used to target different subfamilies of bromodomains. So we set up this assay for BRD41, and then you can see that it gives you a pretty good assay window, and the signal is very high. And then we use this assay to screen a number of in-house libraries. So in total, it was over 68,000 compounds that we screened. And then the Z prime factor was about 0.7, so it's a pretty robust assay. Our hit rate was from 0.1 to 1%. And then what was very exciting is that we were able to identify novel chemical series, and then the compounds were BET specific and showed low micromolar affinity. So here's an example of screening BRD4-1 screening statistics shown on this screen for Cambridge and Maybridge libraries, particularly the hit rate on the left, and then the assay window on the right, and in the middle, which show how many hits were sort of selected based on their compound properties to take to the next level. So this is some screening capabilities that we have at RBC. Oh, sorry, I forgot the compounds were tested using a thermomelt assay for profiling, using selected members of bromodomain subfamilies, and then you can see from the heat map that the current lead compounds are specific for bed family. So we also known for our screening profiling with radioisotope assays, and even though there's so many different technologies being developed and sort of steering away from radioisotope use, but it's still considered the gold standard for profiling in HTS. And then we use radioisotope assays for kinases, PARPs, methyltransferases, HATs, to name a few. And one example is profiling for methyltransferases in our RBC hotspot. And this is a miniaturized radioisotope filter binding assay, and it measures total methylation of protein or DNA. And you can use any substrate from nucleosome to peptide in the same platform. It's compatible with HTS and mechanistic studies. And on the left on the tree, the circles in green indicate which proteins are included in the panel. And on the bottom, you can see a reference curve for NSD2, the control compounds, and this kind of data is included with every protein that we test at RBC. Obviously, not all proteins are compatible with radioactive platform, and so we try to use other very sensitive technologies to ensure that the assay window is high, and also that the assay is compatible with potential screening throughput, and such as fluorescence, HTRF, FP, alpha LISA, alpha screen. Also, we're trying to expand our assay set for measuring binding interactions. So currently, if the protein doesn't have any activity, we look at interaction between protein and peptide via alpha screen or HTRF, and that's especially applicable to binders. And then also we can look at protein compound interaction by either DSF, which is same as thermomelt, and then also we have ITC capabilities, and we're very excited that soon we'll be adding SPR, a biocore machine, to our assay selection, and that will be a great complement to what we already have in the lab. We also perform custom assays, so if you have a target that you would like to test, we can take it all the way from through our assay development pipeline by just figuring out initial activity, and then also determining KMs, and then if you're interested, it can go all the way to HTS applications. But we can also modify our existing assays to meet your specific needs, such as use alternative substrate and concentration, incubation time, etc. And then every assay that we perform is compatible with MOA studies to make sure that you know if your compound is substrate competitive or else. And then having this great biochemical panel, we can easily profile the compound either in terms of specificity on the family-wide scale, or also run a full biochemical panel to identify any off-target interactions that can be of great value for further SAR. And then finally, I'd like to acknowledge everyone at Reaction Biology and that make the company run smoothly, and also our funding from SBIR, and then our clients, and then thank you for your attention. And then I hope I convinced you that we have a great suite of epigenetic services, and then hopefully we'll be able to work with you sometime in the future. And then please visit us at booth 21 or visit our website for any additional information. Thank you.